Hi everyone, my name is Karen Whitman and I would like to welcome you to the Organoleptic Analysis, How Our Sensory Panel Can Sell More of Your Products webinar. It is my pleasure to introduce to you Mr. Guy Rimby. Before I do that, I am going to go over a few housekeeping materials. The first thing, this webinar is recorded and the recording will be available in 1 to 2 days. All questions will be answered at the end of the presentation. However, if we do run out of time, you will get an email with an answer to your question here within the next day from Mr. Guy Rimby. And in order to have your questions answered, be sure to put them in the queue. And in order to do that, you'll just go down to the questions panel at the bottom of your webinar screen and click on that, and then you'll just type in your question into the queue, and he will answer them at the end. It is now my pleasure to introduce to you Mr. Guy Rimby, the Project Manager of Stability and Sensory for the Eurofins Nutrition Analysis Center. He has been with Eurofins for 6 years in different capacities of quality, safety, stability, and sensory management. Mr. Rimby has been involved in the development and coordination of approximately 900 shelf life studies. Mr. Rimby graduated from Drake University with bachelor's in biochemistry and molecular biology. The floor is yours, Guy.
Good afternoon, everyone. That is, I guess, if you're mountain and eastward. So what we'll start off with, I wanted to talk about how the sensory panel can help sell more of your product. So it is essential, basically. It's part of the different aspects of shelf life that you want to look at. So even if your product is shown to be completely safe from a microbial standpoint and food safety standpoint, and you still meet all your nutritional claims at the end of your proposed shelf life, and your product integrity is still there, one of the things that final thing that you want to make sure is still great with your product is the sensory portion of it. How are your potential customers perceiving your product? So by having a certificate of analysis that you can rely on for the proof that your sensory meets your expectations and is acceptable, that is that final step in a shelf life program. It also is something that can be utilized from a quality control standpoint and can be used from a batch-to-batch verification that the product is still meeting initial criteria as well. So with that part being said, some items to consider when you're thinking about sensory analysis then is how you go through to train your panelists. For Eurofins, what we went through is an extensive qualification period, and then the individuals who passed that qualification then were further trained. From the actual performance of sensory itself, you need to think about, do you want it to be reported in a consensus manner or independent? So what that means is when you have your analysts sit down to take their observations, do you want them openly discussing it amongst themselves to come up with one collective result, or do you want them separated with partitions or whatever else you have on hand to separate your panelists? And do you want them observing it independently so that you're getting a collection of basically multiple data points from that same time point? Next, how would you like it reported? So descriptive or discriminative are the two ways to look at that. And what you're going to get from a descriptive analysis is words. So if you're looking for terms, if you're wanting a summary of everything to be put into a paragraph form, that's what you're going to get with your descriptive analysis. With your discriminative analysis, what you're going to get is that's where you see those hedonic type scales come in, your ranking scales, where something could have a 1 to 5 scale, 1 to 7, 1 to 10. Really, whatever you take the time to predetermine as the scale that you're going to use to discriminate on whether or not your product is acceptable. And then finally, what aspects are you going to monitor? What aspects are important to you? Is it just flavor and odor? Do you care about the color? Is texture important? We can go further beyond what I would consider the normal four, which is the texture, visual appearance, which is usually observed as color, as well as your odor and flavor. And then some clients will go even further than that. You'll look at things like stratification, consistency, packaging. These are all aspects of a product that we can observe and take note of and develop protocols for that our analysts and panelists can then train on and assess for you. So as I said, I mentioned that there's the four basic aspects of sensory that we look at here at Eurofins. So our panelists, for our panelist training, were assessed on those four aspects. And those four aspects being texture, odor, color, and flavor. For the texture, odor, and color, what we did was a triangle test. And the individuals who went through the qualification process were given two like samples and one different sample. And these individuals then had to be able to go through and assess the odor and color differences and pick out the correct sample that was different in that triangle test. For the flavor qualification, it was much more intensive. Our panelists were given a allotment of 25 different sample cups of different dilutions of all 5 flavor profiles. So we had our sweet, salty, sour, savory, which some people may refer to as umami, and bitter. And what we did was we diluted them all at 5 different concentrations, and then what we made those dilutions with then were distilled water mixed with sucrose for the sweet, sodium chloride for the salty, acetic acid for the sour, MSG for the savory, and quinine for the bitter. And then our panelists were then scored on how many they were able to correctly place in this grid matrix below, and the ones that were shown to have passed the triangle test for odor and color and texture, and also were able to get a certain number correctly placed for the flavor, were then selected for the panel, and we went through with further training after that. So once you have a trained panel, that's when you can start to move on and think about what type of analysis are you going to perform. So when we think about consensus analysis, as I said, this is where all analysts interact together. So this type of interaction is usually done at some type of roundtable where all the analysts can equally assess the product together. They have an open discussion, and they come to conclusions together on how they're perceiving the product. These are usually done in small groups. We usually use 2 to 3 analysts for that, unless our clients have a larger request, and then at that point we would use 5 to 6 analysts for requests like that. Moving on to independent analysis, there are 2 types of independent analysis, and well, technically you can use the descriptive and discriminative analysis as part of the consensus as well. But when you think about independent analysis, the descriptive and discriminative is usually the way that we branch off to select which type you want performed. With the descriptive analysis, these are an analysis where for the independent analysis, the analysts will not interact with each other. These results are collected and summarized by the study director, which in most cases is myself. At that point, the results are recorded using words and phrases. So what will happen is, say we use a panel of 5 to 6, I'll collect 6 different observations based on whatever aspects are requested. So we could have a client request texture, odor, and flavor. We would take each of those aspects. I would summarize each aspect as reported by all 6 of the analysts, and then what the client would receive on the report would be a paragraph summarizing what all of the analysts collectively said. So I would take note basically to really highlight the things that were seen across the board. So I would say things like 5 of 6 analysts noted blank, and then maybe at the end of that paragraph I would list any singularities. One thing that's really important with the descriptive analysis is that if clients have buzzwords, basically, that we get that nomenclature pre-established beforehand, before we start the study, so we can get a protocol developed for that. And one of the things that comes to mind for that is we had a client in the past where we didn't fully get the protocol developed to the level that I would have wanted it, and we had an issue where we were reporting something as pale yellow, but the client wanted it as off-white. And when we showed the picture to the client, they agreed that that was exactly what they had perceived as off-white, that we were perceiving as pale yellow. So when something is very close on that teetering on subjectivity, it's really important to have those buzzwords put in so that we can, and to predefine those with preliminary testing and observations, so that the panel can basically build their own log of what is normal. Here's an example of what you can see for when you're looking at appearance and odor and flavor grades. This is the type of thing that we would use with our clients to pre-establish, to build up a word bank basically for what was acceptable, and then as they went from the top of the page to the bottom of the page, these are their terms going from most acceptable, their baseline levels, to what they would perceive their product to degrade to, and where it would be finally considered unfavorable. On the other hand, if you're not going to do descriptive analysis, the other option then is discriminative analysis. Again, this is a type of independent analysis where the analysts do not interact with each other. These results are, again, collected and now calculated by myself. So instead of going through the summary of making them into paragraphs, what I'm doing instead is a data collection in order to take averages of the numbers that are being tabulated. What's going to be recorded, the analysts are then are recording numbers and rankings. So these numbers and ranking systems are going to also be pre-established in the development of a protocol. So this is where, when I mentioned the 1 to 5 scales, the 1 to 7 scales, the 1 to 10 scales, this is where that would happen. And what would be developed during that stage, during that protocol rating stage, is, you know, 10 is a benchmark product. What does a 10 stand for? And that's what I'll go into more detail on the next slide. Last thing I wanted to mention for the discriminative analysis is when you're talking about the calculations involved with the reporting for discriminative analysis, that is where a triangle test would fall as well. Both of which, the ranking system and triangle tests, are going to be things I address in further detail. Moving on to the triangle test. Triangle test is a type of analysis where analysts are presented 3 samples, 2 of which are identical and 1 is different. And then the analyst must identify the different sample. So for the purpose of when we qualified our analysts, we really wanted them to be able to differentiate which one was not the identical samples. Now, one way that we could use a triangle test in a sensory fashion for actual testing, and what we could do with client products, is say product A, sample A in this scenario is your benchmark baseline product, and sample B is one that you had in your warehouse for 12 months. And you want to make sure that there's no detectable change. So what you would do is we would send those in, we would set up a triangle test, and what you would hope is that there's no statistical difference with our analysts not being able to distinguish A and B, because that would be the goal here, that your month 12 would still be perceived just as well as your benchmark product. Now, the higher that the panelists were able to identify that 12-month product, then you know there's a higher correlation to that being dissimilar to your benchmark. We can also do that in a comparison analysis as well, where you could take product A could be your company's product, and product B could be a competitor, and you could do it that way to see how similar competitive products were to each other. If there was no real statistical difference noted, then we could assess then that the competitor's product and your product are, from a sensory standpoint, unable to be detectable by the general panelists. So those were the types of tests that we would perform here for sensory. I wanted to further break down how we look at those aspects that we're looking at when we're doing these different types of tests. So one type is the visual appearance, and all this stuff that you're seeing now is, and all the descriptions that you're seeing are excerpts from training that our panelists have gone through to be able to better identify and better assess the aspects of sensory. So there's two categories for visual appearance, and the two things that we're going to look at is light interaction and physical form. So with light interaction, we've got four categories. So the lightness, the color, purity, and gloss of a product. So this has to do with how light, is it shiny? Is it green, red, purple, whatever? That all goes into how the light interaction happens. The physical form section of it, then, when we're talking about visual appearance, is broken into three subcategories of shape, surface texture, and consistency. So it could be things with products like, is it runny? Is it thick? The shape, is it a capsule that is 2 centimeters long and 1/2 centimeter wide? Does the surface texture have a grittiness? Is it smooth? These are all things that we would observe as part of visual observation. And again, it doesn't mean that we're going to always observe every single one of these categories. This is all part of that protocol writing stage of which aspects of this, which categories do you want us to include when we are observing the product. Now, if basically we're given a blank slate, we're going to look at this from every aspect and try to pick out which parts we think are most pertinent. Something that a lot of people don't always think about, which is the fifth sense, which the touch somewhat comes in, or the fourth and fifth sense would kind of go hand in hand with touch when we're thinking about how we perceive a product. So touch has two main categories. We have the finger feel, and then we also have mouth feel. And with finger feel, what we're mainly looking for there is the firmness and texture. So again, from the visual aspect, we can look at something and say that it looks smooth or that it looks gritty, but then we confirm that with the finger feel. With, yes, it actually is feeling gritty or smooth. And then we also look at it from a firmness standpoint too. So, is it mushy, or is it firm? Is it solid? Does it have some give to it? And this would be a section of all this, everything I've been mentioning is we can determine the exact nomenclature during the protocol writing stage. So if mushy is a failure, basically, if that's the word that you want being used for when your product is no longer considered acceptable, we could use that. If you have a different word for that you want to use, that would be all part of that protocol writing stage. With mouthfeel, what we're going to try to detect as we are tasting and chewing the product, is there hardness, brittleness, chewiness, grittiness, fibrousness, moistness, greasiness. These are all the types of words that we're thinking about and would be utilizing when we think about how we are perceiving the texture of a product within our mouths when we're assessing it. And the fifth sense that I wanted to point out then is hearing. People don't always think about hearing as one of the senses that's associated with sensory of a product, but for some evaluation, this can actually be a critical point for them. You know, if you're looking at something like a soda, for instance, you know, is it fizzy? Maybe it's cheese curds. Do they still have that squeakiness that you are characteristic with them? Or is there like actually a crunchy sound for some of the products, like say a rice cake or something like that? Odor would be basically the third sense that we perceive, with basically hearing coming in there at our honorary mention fifth. After we look at the visual, then the touch, and then we've got odor as a third. This is basically the order in which our panelists will assess the product. When it comes to odor, odor is perceived not only as we breathe in, which is your orthonasal perception of odor, but as well as when we breathe out, which is your retronasal perception of it. So there are olfactory nerves that are found at the very back of the nasal passage. So what we do to make sure that we get interaction for those is we make sure our panelists are taking the aromas in by breathing through their mouth and then exhaling through their nose, while also taking that orthonasal perception by breathing in through their nose and really inhaling that odor so they can get a full perception of what the product is producing. We will also use wafting to increase air movement, and that will again help intensify the perception of any aroma. Odor plays very heavily into flavor. The perception of flavor is the combination of taste with odor, and smell actually makes up 85% of our flavor experience. So one thing that I always stress with our panelists is, you know, we schedule these panels at least a day in advance so that our panelists can adequately prepare for them, because it means not putting on scented perfumes and lotions, because those types of additives to the body can really throw off a sensory perception for odor and flavor. Finally, there's taste, and taste is always the last aspect that we look at when we're perceiving a product. And taste is divided into those 5 stimuli that I mentioned earlier: salty, sour, sweet, bitter, and savory/umami. It's very important that the palate is clean for this part of the perception. So up to an hour leading up to the evaluation time, the panelists are knowledgeable that they should not have any mints, gum, coffee, or any other food that might interfere with their palate being clean. There's 3 stages of taste that we will detect as well. So you know, some clients just look for an overall summary of it. Other clients will really want the stages of taste broken down, and how we'll do that is we'll take an initial taste, the evolution of taste, and then finally the aftertaste that is left in the mouth after tasting the product. So one of the things I wanted to point out too was just the perception of different thresholds. If you go back to when I had that chart of the different dilutions, you can see that on this graph here, the quinine, I'm going to try to bring the mouse over here, the quinine right here has a perception at a lot lower concentration than what the sucrose is. So if you were to deal with equal dilutions, the quinine is going to interact with the palate much more readily than sucrose will. So that's something to keep in mind when you're putting together if you're putting together a dilution panel for qualifying panelists, is that if you do everything equally, the bitter is going to basically be perceived much quicker than the sweet that you see here or the salty. Further training that we talked about with the analysts is exactly how to taste. So what we want to talk about is, how do we describe those first sensations of initial perception? So that's where we get that initial flavor, that very initial perception that you get as the product first hits your taste buds. Then before we really describe the evolutionary part of the flavor, what we want to do is we want to make sure that we fully masticate the sample. So chew it up as much as we can, allow saliva to mix with it, and we're going to do it in a way that allows the sample to fully coat the tongue. So if we're dealing with a liquid here, the chewing part would not be necessary, but we're really taking in enough so that we can fully coat the tongue, and then we're going to try to push that sample against our upper palate with the tongue while we breathe through the mouth, allowing air to contact the tongue, because again, this is going to help increase the perception on our palate. And that's when we would then note the evolution flavor stage of taste. Finally, we would continue to note any latent perception that would happen after swallowing or removing the sample from our palate. So, if we decided to expectorate the sample slash, that's a fancy science word for spitting it out, then that was where we would get the aftertaste there. So the last stage of this webinar that I want to talk about is what types of reactions the analysts have been trained to identify. So when we are tasting, we've gone over some different types of degradation products that they look for to help identify from a sensory standpoint when a product is degrading and going bad. So oxidation is one of those. And what oxidation is it's the reaction of when oxygen has access to products that contain fats or pigments. What oxidation will do to a fat is it makes it become rancid. So if you've ever had a really old bag of potato chips, the oils on those chips will start to go bad. You get that rank odor, that rank flavor, because what's happening is the triglycerides are being hydrolyzed into free fatty acids. And another thing that can happen is the unsaturated fats in vegetable oils and things of that nature, their double bonds will start to cleave and be released as aldehydes and ketones, again, making that rank odor and flavor. Another thing that they note, less from a flavor standpoint, but more from a visual observation, is changes in pigment. Color changes are really important because as the product will oxidize, the colors will change. So for example, you could have a nice red, fresh ground meat that starts to turn grayish brown as it gets oxidized. Avocados, they can go from a really vibrant green to brown. So these are other things, those are examples of color changes that we would note as well with oxidation. One of the things that comes into play with oxidation is rancidity, as I mentioned, and recognizing early rancidity is important because late stage rancidity is easy to detect. It will get such potent, unfavorable flavors that any general consumer could pick up on that. So what we're trying to do is we're trying to detect those types of notes that are characteristic with rancidity before it gets to that late stage. So what the analysts have been trained on is they're looking for aromas that are similar to that of crayons or putty, a mouthfeel that leaves a greasiness that was otherwise not there from the beginning. And also from a visual standpoint, looking for slight darkening in the color of the appearance. So if any grain, browning, any type of darkening happenings, that's where they're looking for the rancidity early stages in a visual aspect. And then also it can lead to a change in texture. So something that was once crisp or hard can become pliable or soft, and then the mouthfeel can become gummy or almost gritty sometimes and stick to the teeth. Another the aspect of staling, which is sometimes confused with rancidity. The big difference between rancidity and staling is rancidity is the breakdown of the fats, where staling is the chemical and physical process of the reducing of the palatability of starches and carbohydrates. So we're looking at a different group in the nutrition here. So we go from fats to our carbs now. What happens during the staling process is moisture is migrating from starch granules into the interstitial spaces and degelatinizing the starch and causing a recrystallization. So that's what's causing textures to become leathery or hard when something becomes stale. So if you've ever had something like a rice cake that sits out opened and it becomes it loses that nice airy crispiness, that's because we're getting a staling there, where we're getting that recrystallization of the starches that's making it that hard, leathery texture. The other thing that can be lost to the air during the staling process is that fresh flavor. So during the staling process, what will happen is taste will take on the characteristics of packaging and storage conditions. So if you have something open, for instance, in an area and it's not being kept airtight, vacuum sealed in some manner like that, and you've got it with other really fragrant type products, it can start to take on some of those flavors during the staling process as those starches break down and then reform, recrystallize. Fermentation is another aspect that we take note of. And what fermentation is the conversion of carbohydrates, usually sugars, into alcohols and carbon dioxide from the presence of yeast or bacteria. So one of the things that we'll note if we think that fermentation is present, is one of the first things we'll see is a bloating of the packaging. So that would be something that we can note even if you don't have packaging requested as one of the aspects of sensory. From an aroma standpoint, we're going to try to pick up on those notes of alcohol that are characteristic of fermentation. So our analysts have had a chance to smell what that aroma is like when you're getting those notes of alcohol. And then finally, when it comes to the flavor perception, is are we picking up sour notes? Because that can be indicative of fermentation. So that's how we would note the fermentation aspect. So now that we've kind of talked about the fats and the carb breakdown, the last thing that you would look at is the breakdown of proteins, and that's synuresis. And what's happening here is it's the release of moisture contained within protein molecules. So what happens when we see synuresis happening is we're going to see visual cracks and fissures in the product, creating a pooling of liquid. Something that's really characteristic of that is if you have something like a cream cheese, for instance, you can start to see that fissure and crack as it ages. Moisture will be released, and that's part of the synuresis. And what's happening during that to their aroma and flavors, it's becoming less potent because you're having some of those proteins that would otherwise aid in the baseline type aroma and flavor perception are being lost in that synuresis process as the proteins break down. So outside of your fats, carbs, and protein breakdown, we also look at pungency. Regardless of what's causing the pungency, we just look at it from what is, we look at what is the strength of a smell or taste present in a food. And pungency is not sensed by our taste buds, but instead from the stimulation of our somatosensory fibers in the mouth and tongue. So as things become less pungent over time, it's because of the loss of volatile oils that evaporate off or become oxidized. So somewhat still part of that whole breakdown of fats, but perceived in a different way. So this is important with anything that has herbs and spices added to it. Pungency is a huge part of those types of products. or any other types of natural artificial flavoring as well. And with that, we've kind of run the gamut on you know, the different ways to put together a sensory panel, the different types of panels, of whether or not we're looking at a consensus or independent analysis for gathering information, the type of information gathered, whether it's descriptive or discriminative, and then finally exactly how we go about perceiving all those aspects of sensory, including to but not limited to texture, color, aroma, and taste. Thank you guys all for your attention. I'll answer any questions that may have come in during the webinar at this point.
Thank you, Guy. That was very informative. And now, as you guys get your questions in, I will go ahead and ask them. So the first question is there anything we should pair our analysis with, like other testing or anything like that?
So one thing that Eurofins will require is that we have some proof. If you're just going to do a sensory analysis in the aspects of only texture, color, and aroma, we will not require that it's really paired with anything else from a mandatory standpoint from Eurofins. But if as soon as you require taste as part of those aspects of sensory, we will require that you have microbiology performed on a preliminary sample so that we can see that the product is safe for our analysts to consume. Now, if you've already had microbiology performed, you can also just supply a certificate of analysis to us to show that the product is safe to consume, and we will go forward with the taste testing portion of it in that sense. Now, that's just for whether or not how we would go about taking on the project. Now, if you're looking for other types of testing to pair with sensory, if you're looking at it from a standpoint of shelf life, I would say it's important to look at, you know, food safety with microbial testing. You know, you're looking at aerobic plate count, yeast and mold, and other pathogens. It's also important to look at your nutritional claims, so whatever you have on your label. So if you have any vitamins or other bioactives that you look at those as well. And then other parts of just the physical integrity of your product. So what's the pH, what's your moisture content, what's your water activity levels look like. Those are all important from that standpoint. And then if it's not for a shelf life, but just a general quality check, I would say, you know, establish some threshold values of what you're looking for, and then have those assays performed alongside sensory, just to make sure that from a general one-off standpoint, whenever you're submitting a sample for general quality checks, that you've got others testing tied on to your sensory.
Thank you, Guy. We actually don't have any more questions at this time, so we'll just wait a couple minutes, and if anyone comes up with a question, please go ahead and put it in the queue. Like I had explained earlier, just click on the questions section of the webinar panel, and then click on the text box, and then type your question in. Okay, we actually now have another question. The question is turnaround time. Can you discuss maybe how long it takes for different types of products?
Definitely. So when we're scheduling our sensory analysis, we will request that our clients give us basically a one-day heads up before they send the product in. And the reason we want that is because we're going to take those 24 hours to schedule a sensory panel assessment. So what that 24 hours will allow us to do is to get, you know, 3 or 6 panelists, you know, 12 panelists, whatever the number of panelists that the client's requesting for that sensory analysis, we will get put together and have them set aside, so that way they have the time to the 24-hour lead time in order to get together and meet. Now, once the sample comes in, we will test that sample within 24 hours of submission, and the turnaround time on that after that is the total TAT is 7 days. So we will actually test and perceive the sample within the first 24 hours of receipt. However, you know, for some of those descriptive analysis, it does take some time to get everything typed up and put together for a final report. So that's where that extra time happens. It's not that the product is actually sitting for multiple days waiting to be tested. It's going to be tested within the first 24 hours, but that 7-day turnaround time comes in the actual typing up and drafting and reporting of the results that are observed.
Thank you. That is all of our questions for today. So we're going to go ahead and conclude this webinar. Thank you again for all of your insight, Guy, on sensory panel testing. Everyone, please look for an email here in the next day with a recording of the webinar along with the PowerPoint slides. Thank you again, everyone, for attending, and Guy, for your time.
Thank you.